All right, welcome back to part 2 of chapter 26, Phylogeny and Tree of Life. So we kind of ended with the part 1 about this video where he was showing you how to read a phylogenetic tree, and then he asked was the closest relative to cheetahs. So I forgot to go over the last part of the video, which is about whether these two representation of the phylogenetic tree are they showing different relationships. And I want to go over this with you really quickly because we talked about how phylogenetic trees can be represented in 3 different ways. So here is a very good example of representing two different two representation of the same phylogenetic tree. So he asked, do these two phylogenies show the same hypothesis? So here is this branch point that goes to bears and this whole group of species. Here's this branch point. I'm going to keep dotting the branch point. The cheetah with this group of species. Here's this branch point that goes with these two tigers, or this branch point that goes with sister taxa, lion, and leopards. So in this phylogenetic tree representation, are we looking at the same thing? So let's put in the dots and see how it goes. Here is one branch point going towards the bear and the rest of the group. Here's another branch point that goes toward the cheetah and the rest of the group. Here's another branch point that goes towards the tiger and the rest of the two groups right there. And here's that branch point that goes between the two sister taxa, lion and leopard. So regardless whether it's represented this way or this way, the relationship as I have just shown you still is the same. Okay, so as long as they have two taxa that share the same common ancestor on either representation on the phylogenetic tree, then they are equivalent to each other. It doesn't have to be a particular order. Like I said, here you can see that you have tiger at the top and then bear at the bottom. On this phylogenetic tree, you have bear at the top and the cheetah at the bottom, and they actually kind of mixed up the order in which the lion, leopard, and the tiger goes. So this is why I said the order in which these species are portrayed on the phylogenetic tree does not matter. It is the evolutionary relationship that they share, that one common ancestor. That is what's important from a typical phylogenetic tree. Okay, we will talk about, like I said, phylogenetic tree that has exceptions. We'll go over that later. All right, so here are 3 key points to remember about interpreting phylogenetic trees. Remember, interpreting them correctly is the key, because as you know, the speaker was talking about in the previous video, is that even graduate students can make a lot of mistakes and confuse themselves and read the phylogenetic tree wrong. So this is why I want to emphasize how to interpret it correctly. First, they are intended to show patterns of descent, which is the common ancestor between 2 lineages, is all it's trying to do, not phenotypic similarities. Okay, what do I mean by that? For example, crocodiles are actually more closely related to birds than lizards, but they look more like lizards because morphology has changed dramatically in the bird lineage. So if you look at this particular phylogenetic tree right here, here are birds. Okay, birds, here's crocodiles, okay, and there's lizards. So technically speaking, birds are actually more closely related to crocodiles. But by morphology wise, we would say that lizards, they look more like lizards morphology wise. Okay, they have scales, etc. You wouldn't say a bird looks like a crocodile. No one will say that. But when we look at the genetic lineage on the phylogenetic tree, they say birds is actually more related to crocodiles than to lizards. So that's why when we're looking at these evolutionary ancestral relationships, we're not looking at how similar they look. Okay, that is the morphological, morphology homologies we've talked about earlier. But for phylogenetic trees, we're purely looking at, what designate this particular lineage as this became this became diverged to become that, etc. Sometimes is traits, and not only just structure, sometimes it's traits as well, and sometimes is genetics, DNA sequences that we'll talk about later in a little bit. All right. So the second thing is that we cannot necessarily infer ages of taxa or branch points shown in a tree. So what do you mean by that? For example, a tree does not indicate that this particular tree right here, phylogenetic tree, does not indicate that chimps have or chimpanzees have evolved before humans. There's no sequence, or vice versa, that we have first, somehow we have the first chimpanzee here, and then we have the first humans here, and the humans, chimpanzees came before humans. Well, there's no way to tell that on a phylogenetic tree. All phylogenetic trees show us is that they share the most recent common ancestor, which is right there at that branch point. It does not deal with anything or says anything about the sequence of each taxa, which one comes before the other one, or because that will be a problem if we represent the phylogenetic tree diagonally or vertically, because then things become out of order, or if we rotate about the branch point, then that will changes the sequence. So general phylogenetic trees does not infer ages of each taxa unless otherwise indicated. We'll talk about, like I said, I'll talk about those exceptions in a bit. Third, when we're just talking about general phylogenetic tree things. So third, we should not assume that a taxon have evolved from the taxon next to it. What do we mean? So again, going back to this tree, phylogenetic tree. This tree does not indicate that humans have evolved from chimpanzees or vice versa, or that chimpanzees have evolved from humans. It only shows that we share, okay, chimpanzees and humans share a common ancestor that is now extinct. And that common ancestor is neither human nor a chimpanzee. Kind of like what the speaker in the previous video talked about, is not that there was this, this this leopard became a tiger. It's just that they share the same lineage, where there was this common ancestor that was neither a tiger or a leopard, and then they have somehow diverged and became two different species. Same thing here. You have this common ancestor that was neither a chimpanzee nor a human, but they have diverged to become chimpanzees in this lineage and became humans in this lineage. Okay? So that's what the three key things about interpreting general phylogenetic trees. And like I said, there are exceptions. I'll get to that in a bit. Now, before we continue with the next section of the chapter, let's quickly do a few practice questions about interpretation of the phylogenetic trees, as I know, it can be confusing at first. So let's go over these three questions really quick before we move on. So let's look at this question. Based on phylogenetic tree shown here, which of the following statement is true? Species C is mostly closely related to D. So they're saying this. They're mostly closely related or sister taxa of each other. Species D originated from species C? No, probably not. We said we are not saying that one evolved into another on the phylogenetic tree. So that goes nope. Species A is most closely related to species C. So here's species A. Remember, if we're talking about the closest relative of species A according to this phylogenetic tree, the whole part here or group here is equally related to species A. So species A is not more closely related to B, C, or D. Species A is actually equally related to B, C, and D. So nope, that is incorrect. And then last statement, species B is more closely related to species C than to D. Again, now we're looking at species B. And if we're talking about phylogenetic tree, here's a common ancestor. Okay, so then species B is equally related to species C and D, not more closely related. Just because they're next to each other like that doesn't mean they're sister taxa. We're going to come back here to look at where the branch point holds, okay? C and D is sister taxa, most closely related, but B is not more closely related to C than D, so that is incorrect as well. Hence, the answer is the first one. Species C is most closely related to species D as your correct statement. Okay, so the next one, I'm going to let you do it while I read the question. So make sure you're ready. The branch point marked with a red dot right here on this phylogenetic tree represents which of the following statements? You can pause the video, do it yourself first, and then unpause it to listen to my reasoning and answer. So the first statement says the only common ancestor shared by species C and D. Is this the only ancestor? No. Remember we said that this horizontal line here represents many, many different ancestors that have eventually become your species C. So that is incorrect. The only is incorrect here. The most recent common ancestor shared by species C and D. Yes, because this is the branch point that splits into C and D. So this is a correct statement. Let's see the other two. The member of species B that gave rise to species C and D. So the one of the common ancestors that gave rise to B is not this red dot here. It is this right here. I'm purposely drawing a star so you don't say, oh, that's two dots. But this particular star is then the common ancestor for species B and C and D. So that's why this one is not the common ancestor gave rise to B. Member of species C gave rise to species D. Again, going back to phylogenetic tree does not say anything about one species evolving to become another species on the spectrum. Okay, so C did not give rise to D. Okay, they are just close relatives of each other or sister taxa of each other. So hence the answer should be the second statement. The most recent common ancestor shared by species C or D is indicated by this red dot right there. Okay, next one. According to this phylogenetic tree, what can we say about species K? Go ahead and pause the video and then unpause after you're done reasoning yourself through. Okay, so let's go through these statements. It says that species K, which is right here, is most closely related to H, right here. Incorrect, because species K, if we trace back to its common ancestor, is actually most closely related to L, M, and N, not more or less. Okay, they're equally related to K. So the most, as this was not a correct statement. So next statement is as closely related to species L as it is to species N. L to N, as closely related. That is correct. Third statement is more closely related to species L than it is to any species. Incorrect. Remember, we just said K is equally related to L, M, and N. So that is not correct. And that K originated from species J. That is also incorrect. K does not originate from J. There's no evolved sequence here shown by the phylogenetic tree. Hence, the answer is then the second statement is correct. That K is closely related to L as it is to N, as it is to species M as well. Okay, there's not more or the other, is equally closely related. These three are equally closely related to K. All right, so now that we are, feeling pretty confident about the interpretation of phylogenetic trees, we can discuss the real life application of phylogenetics. Because you're like looking at this, is like, how is this related to me? I'm not going to become a systematics one day. Well, who knows? You may have, a job that will eventually maybe use these phylogenies. I don't know. The fact that you're taking biology for majors, maybe you're interested in science. So here are two real life application we can use phylogenies for nowadays, okay? So one application is that we can find organisms that are close relatives of an important agricultural crop and serve as reservoirs of beneficial alleles. What does that mean? Well, let's say, for example, corn is now an important food crop worldwide, not just in the US, but in the whole entire world. So, based on DNA data, researchers have created a phylogeny of corn and identify two species of wild grasses that may be corn's closest living relatives. Okay? Now, knowing that gives us very helpful information. Let's say if somehow corn was ever became scarce, that we don't have corn anymore, or somehow they become extinct. Okay, you don't know. Maybe somehow in the future there's this natural selection or genetic drift of some sort that has caused the corn to become now close to extinction, just like those great prairie chickens we talked about in the last chapter. So beneficial alleles from these two close relatives actually can then be transferred to cultivated corn by crossbreeding or genetic engineering. Let's say there's this specific species of corn that is so great and helpful to the world, but yet we're losing them. What can we do? Are we at doom? Okay. Knowing phylogenies of corn, then we can say, oh, we have these two close relatives of this specific corn that we can get those beneficial genes from, because the fact that they are close relatives means that they have similar DNA sequences. Okay. So if they are close relatives, it very likely means they have very similar genes. And if this particular gene is very important for this special corn, we can't just take any old corn and use genetic engineering techniques and put this particular beneficial gene into those corn so that we can, in a sense, engineer our own specialized corn. Okay, so that's why phylogenies can be an important usage or application in this sense, in the agricultural sense. Okay, and in the same way that, right now, I think people are trying to clone mammoth, cloned species that are now extinct. And, I'm sure the way they do it is something to do with phylogenies. I haven't looked into that, but I know there is research companies out there nowadays that are trying to clone animals that were once extinct. I don't know how successful they are yet, but I think this is going along the same lines of using phylogenies to do that. I don't know, but that is one thing that I have heard of. All right, so another application is that we can actually infer species identities by analyzing relatedness of DNA sequences from different organisms. So one example of this usage is that we have researchers that have used this approach to investigate whether whale meat that are sold at Japanese markets have been harvested illegally from protected whale species, and these species are specifically protected under international laws, or are these whale meat actually from species that can be harvested legally? Because whatever whales are out there, who knows what they catch? Okay, what we see as a consumer is that these are whale meat available at the supermarket. Okay, I'm talking about Japan, not in US. In Japan, there is this whale meat, but who knows where that meat came from? So here are these specific researchers. There are names to these researchers in the textbook. If you're interested, go back to chapter 26. There's a whole story about these two researchers, what they did, etc. But they decided to do this project and get samples from the Japanese market, and they try to sequence what species of whales are these mystery whale meat from. So in this research study, 14 samples of whale meat were purchased from Japanese fish markets. They sequenced part of what's called the mitochondrial DNA. Now, we talked about back from 1406, biology 1406, that each cell have DNA inside the nucleus. Well, turns out there's DNA in the mitochondria as well. And that's why they harvested mitochondrial DNA from each sample and compare their results with the mitochondrial DNA sequences of known whale species, okay? Because we have those sequences out there, okay, known, so that, if you're this whale species, you have this sequence, etc. So by international law, only minke whales caught in the southern hemisphere can be sold legally in Japan. Okay, this is by international law. And to infer the unknown species identity, these two researchers constructed what's called a gene tree right here, that is where they are taking genes that from each of the whales, okay, and they shows patterns of relatedness among the DNA sequence rather than taxa. Okay, so this is a different kind of phylogenetic tree, whereas not showing, common ancestry, taxa, etc. We're talking about how related are these species in terms of their DNA sequences. Okay, so that's how they are able to end up with this resulting gene tree. Now, it turns out that only 8 of the 14 samples were actually legally caught. So here are sample 1A through 8. So they were actually whale meat that were closely related to mink whale species from the southern hemisphere, while the other 6 samples, which is the red right here, unknown sample number 9, unknown sample 1B, 10, 11, 12, 13, they are actually 6 of these samples were related to whales that are not legal to harvest from. Now, if no one bothered to spend the time and effort, because this is a lot of work and probably money, to sequence these whale meat sample, no one would have known that some of these markets were doing some shady business. Okay. And on the same note of this story have actually reminded me of a documentary I watch on YouTube about Japan's annual dolphin hunt. This also has to do with Japan, and it is not that I'm discriminating against them. Okay, please be clear that it's just that there's this particular story with that country. I could easily talk about China. I could easily talk about Europe. Whichever the case, but just that this sample brought up this particular idea or application brought up this Japan story. So, annual dolphin hunt is something they do every year. I don't know if you want to watch it, but forewarning is that it can get very saddening and traumatizing. But for those of you that are interested, it is, you can watch it, but I'm not saying you have to. It is up to you, all right, if you want to watch it or not. I watched it. Unfortunately, dolphin hunting is legal in Japan compared to United States. So they do it every year in an area called Taiji. And travelers from other countries have actually exposed these people in the documentary. But like I said, it is legal in Japan to do that. I mean, thankfully, dolphin hunting is illegal in the United States. I just can't imagine anyone hurting those smart and beautiful creatures that are just having their own part in this world of ecosystem we live in today. We have already discussed some examples of human impact on population of species in previous chapters. Remember the pollution we talked about that caused a few species of cichlid fishes? We're supposed to have these nice different species of cichlid fishes, with one with blue back, one with orange back. Now, because of pollution that produce some murky water, you have a few species of cichlid fishes losing alleles. And then we have the story about humans converting, land to farmland that almost killed all the Great Prairie chickens. All those are just, few examples of what human impact have in our ecosystem, okay? And what we call biodiversity. This is why it's important to study biology, because things like this would not be exposed and problems will continue until we lose species one after another. With the knowledge we have today, we can at least try our best to protect our endangered species, because biodiversity is such a critical part of our ecosystem. I really am passionate about the topic of conservation biology, but unfortunately, we will not have time to go over why biodiversity is so important. It is covered in the textbook, but we just don't have time to go over it. But if you are interested, you feel free to read about it in chapter 54 to 56, which are all about ecology, ecosystem, and conservation biology. For now, I really want to show that 1 minute video, but I didn't embed it here. I'll show you it to you in a later, as a separate thing on eCampus. But biodiversity is very important. We can easily destroy it, but yet it is so hard to rebuild the very important biodiversity. I'll show you that video later on. But I have not embedded it here, but that one quick 1 minute video will tell you why biodiversity is important to each one of us, even if you are not a biologist. Okay? All right. So we have discussed before that phenotypic and genetic similarities due to shared ancestries are called homologies. And morphological homology, where it means that they are that we're talking about structures that look similar to each other. And in previous chapter, we talked about them being forelimbs of mammals, and we talked about, I think, the comparing forelimbs of bats and humans and dolphins, etc. That the forelimbs of these animals are similar in their bone arrangements. And then we have what's called molecular homology, which is where we're talking about similarities in genes or DNA sequences. If they should be homologous or similar in sequence if they have descended from a common ancestor. But similarities can also result from convergent evolution, and this is where we're calling it analogies. Okay, homologies are not analogies, nor is analogy homologies. Okay, we'll talk about difference between these right now. Okay, first of all, what is convergent evolution? It occurs when similar environmental pressures, known as similar environmental pressures or natural selection, produce similar or analogous adaptation in organisms from different evolutionary lineages. Okay, that means that you have these two species that are completely under different evolutionary lineages. They're not sister taxa of each other. They're distantly related, but yet they have these similar traits with each other, all due to convergent evolution. At least that's how they explain it. We cannot say that this particular trait is homologous with this species trait because they are not close relatives of each other. They don't have those same trait because of a common ancestor, but not because of sharing a common ancestor. They are analogous to each other, not homologous, because they have similar traits due to being put under similar environments, not because of similar common ancestors. Okay. All right. So, for example, here's an Australian mole, quote unquote mole, and an African golden mole. And they look very similar on the external side, but internally, they have different anatomy, physiology, and also completely different reproductive systems. So these two species are not closest relatives of each other. Their common ancestor and most of its descendants are actually not mole-like. And so analogous traits in these two moles, they have concluded that it has evolved independently due to similar lifestyle adaptation and should not be considered when reconstructing their phylogeny. So just by looking at these two moles, you're like, oh, they look pretty similar. They have these sharp claws right here. They have sort of a pointed head just the same way. So they're closely related. We're going to put them next to each other as sister taxa on the phylogenetic tree. That is incorrect, okay? Because of all those other things that are completely different in these two different moles, that these are just analogous traits. They are not homologous traits. Hence, when we are constructing a phylogeny, we have to be careful of analogous structures because they can easily be a source of confusion. Remember, phylogenies are based on shared ancestry, homologies, not analogies. So another clue to distinguishing between homology versus analogy is that the more elements that are similar in two complex structures, the more likely they have evolved from a common ancestor versus being analogous. Okay, so what I mean by that, for example, let's say we're talking about composition of skull from human and skull of chimpanzee. Okay, the skull, if you've taken anatomy before, is made up of the fusing of many different bones on the skull itself. When they compare the two skulls of human and chimpanzees, they match almost perfectly bone for bone, okay? They both have a parietal bone, they both have a frontal bone, occipital bone, etc. They may shape a little different, but they have those bone structures there. Thus, it is highly unlikely that such complex structures were analogous and had separate origins. Is not likely, okay? These two skulls were highly likely homologous structures inherited from a common ancestor. So that's pretty much saying that for really complex looking things or traits, it is very unlikely that two complex things that look similar to each other happen by chance or analogous, okay? Analogies, but they are actually shared by a common ancestry. Same goes for genes and DNA sequences. The more sequences two organisms' genes share, the more likely that those genes are homologous versus analogous. Okay? So make sure you remember not to confuse between analogous structures, which can be a source of confusion when we're constructing phylogeny. If, let's say, you are a systematist, where evolutionary relationships on phylogeny should be based on homology, shared ancestry, not analogies. Okay, so that's why this section of the chapter goes, distinguishes between the two things. So we have talked about how to compare anatomical homologies, but how do we compare DNA sequences between species, or what we call molecular homologies? We have talked about, okay, we have said that these two particular species have similar molecular homologies, but how do they exactly do that? So this is kind of a glimpse of it. It's a very complicated process, but we'll give you a glimpse of it. First, genomes of species being studied have to be sequenced. You have to sequence the entire genome from the very first letter to the very last letter on all the chromosomes of this particular species. Then we would align comparable sequences from these two species that we're looking at. What do we mean by that? We would put two genes that are supposed to be homologous between these two species and look at their sequence. If the species are very closely related, then sequences will differ at only one or a few sites. Okay, maybe there's a switch of an A here, there's a switch of a GT here, etc. But there's very little of those throughout the gene. If the species were distantly related, then sequences will differ at many sites and may even have different length. Why different length? Well, they say that there can be what's called insertions and deletions that accumulate over time that lead to these species diverging more and more away from each other. So insertion is where we're inserting an extra nucleotide into the sequence, okay? Now, all these terminologies is given that you had 1406 before coming to this class, okay? So you should know what a genome is, and you should know what a nucleotide is, okay? A, T, C's, and G, 4 nucleotides. So the entire genome have all of these 4 is made up of these 4 letters in sequence, okay? And hence, when you're talking about an insertion, it means you're inserting an extra letter into the sequence. Or when you're talking about deletion, you're taking out either 1 or 2 or even sometimes 3 nucleotide from the sequence. All right, so here's an example. Let's say you have 2 species DNA and that their non-coding DNA sequences near a gene are very similar. But the first base of the sequence was deleted in one of the species. This deletion actually will result in an entire DNA sequence shifting back by 1 nucleotide. When we talked about mutations back from 1406, we say that substitution mutation is less detrimental than deletion or insertions, okay? Because when you're substituting just 1 letter, it may be a neutral mutation, it may be just 1 amino acid change. Depending on whether that 1 amino acid can change the final structure of a protein, you may or may not have the disease. In sickle cell anemia's case, we talked about in the last chapter, that was a detrimental thing, why substituting 1 nucleotide. But when you have deletion and insertion, usually it's a very serious case we're talking about because you're shifting back the entire sequence. What do we mean by that? Okay, let me draw it right out really quick. All right, let's say here's A, T, T, G, C, C, A. Let's say an arbitrary sequence of this particular species. And now we're going to take away one of this. Okay, we're going to take away D, G right there. Remember, back from 1406, we talked about transcription and translation, and we said that whatever your DNA sequence is, you will chunk out a messenger RNA. Messenger RNA then goes to the cytoplasm, be translated into a protein, and how those messenger RNA is read is by 3 nucleotide sequences, which is called a codon. So if you had A, T, T, G, C, C, A to begin with, AATT will be your first, second codon, because you have to look to your first AUG starting codon. Let's say the ATT was a second codon. GCC is supposed to be a third codon, and so on and so forth. But when you take out a G, the next codon is no longer GCC. The next codon will be CAA and shifts all the codons after that, okay? This is why either a deletion or insertion of a letter or a nucleotide is so detrimental, because of the fact that it recombines all the codons after the insertion or the deletion. It completely messes up the sequence after that. Okay, so that's why when you have distantly related species, they probably have shared sequences before, but because of insertion and deletion, they have gradually diverged farther and farther apart. Okay, all right, so when you're comparing these two sequences, they may appear very different after a deletion, because you deleted a letter out of this, everything shifted back, you have different lengths of DNA, so they may actually look very different to us by eye if we're just looking at these sequences, these letters, by eye. Nowadays, there's actually computer programs that is used to identify matches by testing possible alignments for comparable DNA segments of different lengths. Okay, so nowadays is pretty advanced, where we have computer programs that can analyze sequences between two species, and they can actually look for possibility of insertion and deletion that can match up closely related individuals or species. All right, so I've talked about it with drawing the letters, etc. So let's see the picture that is given by the textbook. Let me discard those particular drawings I made earlier so it's not confusing. And then looking at this picture right here. In this picture, we have 2 different species homologous DNA sequence. We're not talking about complementary strands, okay? Don't confuse this with complementary strands, or else it would not be same like this, okay? We're looking at this strand of species 1 and this strand of species 2 in a particular gene. And they're identical between these 2 species, letter by letter, okay? Letter for letter. As these 2 species diverge from their common ancestor, let's say there was a deletion of a G right here, and then this G or guanine from species 1 is gone. And then at the same time, there was insertion of 3 extra letters, G, T, and A, in species 2 right here, okay? So if we put in those deletion and insertion, your species 1 and 2 now become this particular sequence. Because of these deletion and insertion, the original orange nucleotides we're seeing in this picture right here that match perfectly before in the original sequence now no longer align with each other. They are actually even of different length. If we were to just look at these 2 sequences by eye, these 2 right here, we would think, oh, these 2 DNA sequences from 2 species don't match up, so they are not related to each other. Now, with computer programs, it can detect anomalies and add space to possible sequence that have been edited by insertion or deletion. Now, after the insertion of these blanks right here, we will realign the matching regions again and show that these 2 sequences are in fact closely related to each other. So they did this type of DNA analysis with the Australian mole and the golden mole, and they actually reveal many differences. Okay, even with this computer program, they still have not found matching sequences like this a lot between these two particular species. So they have concluded that these two species are not closely related. And of course, they did other studies with many other organisms and species and have found those, let's say, the book talks about the silver sword plant, and that they look morphologically very different from each other in the wild. There's tall trees, slender trees versus ground-hugging shrubs on the ground. Turns out that they are the same species, even though they morphologically look different by the eye. Under genetic and molecular analysis, they share many of the same genes. So they found that those two species that are very different by the eye were actually closely related to each other under the silver sword plant family. So that's how we are able to come up with certain relationships on the phylogenetic trees. Not only are we looking at traits, we are also looking at molecular homologies. All right. And in the phylogenetic trees that we have seen and discussed so far, we said that lengths of the tree branches do not indicate any degree of evolutionary change of time. Well, I have emphasized that in the three key things we look for in general phylogenetic trees. But in some exceptions, we have phylogenetic tree diagrams where branch length are proportional to the amount of evolutionary change, or they are proportional to the length of time that particular events have occurred. So in some phylogenetic trees, this can actually be shown, okay? But only if the phylogenetic tree diagram tells you so, okay? If it doesn't say otherwise, you will look at it as a general phylogenetic tree that we have discussed earlier, okay? This is only for cases where the tree tells you that, oh, these branch lengths are proportional to time or proportional to change in DNA sequence or amount of evolutionary change. So for example, in this particular phylogenetic tree right here, each branch length, this represents the number of changes in DNA sequence for that particular lineage, or what we call the amount of evolutionary change that has taken place. So for this Drosophila to have evolved from this original common ancestor shared by all these species, this is the amount of evolutionary change that it took to become this Drosophila fruit fly. Whereas when we're comparing the mouse, it is a lot shorter, okay? Even if we take all these horizontal lines, add it up all together, we're still talking about, the mouse being shorter in length compared to the Drosophila. So the varying branch length indicate that the gene has evolved at different rates at different lineages. If we look at the phylogenetic tree again, like I said earlier, the total length of horizontal lines we see from the tree base to the mouse is still, all of these added together, is still less than the line leading to the Drosophila lineage, or fruit fly as common name. So this means that fewer genetic changes have occurred in mouse lineage compared with the Drosophila lineage. It also then indicates that the rate of change in the mouse lineage of DNA was lower than that of Drosophila lineage. Okay, so this is how phylogenetic tree can show us the amount of evolutionary change that has happened from the roots of the tree, which is this right here, this part right here, root of the tree, all the way to the designated evolved species or taxa. Okay, so for frog, it would be here plus this plus this plus this. Okay, and so on and so forth. For lancet, is this whole thing added with this little horizontal line to get the lancet. All right, so there is another type of phylogenetic tree where branch lengths are actually proportional to time, which means that here you see this particular phylogenetic tree, you have time that is indicated at the bottom here. Where are we getting those time data from? Usually systematists will find them from fossil data that helps to place the branch points at the particular geologic time, at their correct timing. And this particular phylogenetic tree tells us that there is a time that is putting into consideration when we're looking at these horizontal line lengths, okay? So if we look at this branch point right here between human and mouse, that's the particular common ancestor that have eventually diverged to become human and a mouse. This particular common ancestor between human and mouse have started to diverge about, I would say, approximately, of course, this is an approximation, maybe 70 million years ago. And if we look at this particular branch point, this common ancestor between chickens and humans and mouse has started about maybe 320 million years ago. So different phylogenetic tree can give us different types of information. In this particular phylogenetic tree's case, it gives us time. When did this particular common ancestor start to diverge into your final destination species? That's what that phylogenetic tree tells us. And sometimes these can even be combined. When we're talking about, branch length that is equivalent to amount of DNA changes, we can combine it with time, where we would say that these are equivalent to the amount of genetic change, or we can actually put time into these branch length and combine two information into one phylogenetic tree. Okay. So now remember that if no indication was made on the phylogenetic tree saying that this is proportional to time or evolutionary changes, then remember that branches only show patterns of descent and nothing else for general phylogenetic tree. These are special ones that where branch length are proportional to evolutionary change or time down here. All right, the last thing we will discuss about phylogenetic trees is that we should never forget the fact that the way that phylogenetic trees is presented, they represent a hypothesis about how organisms in the tree are related to each other. It's not a permanent saying this is for sure, this has these two taxus or taxa have for sure evolved from this common species, common ancestor. It's just a hypothesis based on what we can know about these species, because there could be things we don't know yet today. So, but based on what we currently know, this is the best hypothesis we can come up with. So that's why they can always change with time, or when more information is known about certain species, or if new fossils were found, let's say. So we can, in addition, we can actually make and test predictions based on the assumption that particular phylogeny or hypothesis is correct. And then in an approach known as the phylogenetic bracketing, this is where we can predict that features shared by two groups of closely related organisms are also present in their common ancestor and all of its descendants, because that's the definition of phylogenetic tree. That's why they're closely related, because they should have these common features, whether it be traits, morphologically homologies, or molecular homologies. They should be similar. That's how they were placed in the phylogenetic tree. So, for example, there is evidence that says birds descended from theropods, okay? And theropods are actually bipedal dinosaurs. So here's birds, here's theropods. So they look like this, okay, for example. So they have come up with this phylogenetic tree as hypothesis to how these species are related to each other in terms of evolution. Again, this is just a hypothesis based on what we currently know and all the data that we have, or fossil records that we have. Here you can see that the closest living relatives of birds, right here, birds are actually crocodiles, okay? Because here's birds, so crocodiles closest relative would be this group of species, which are dinosaurs, which we can't see nowadays. So the only closest living relative of crocodiles are actually birds, and we kind of talked about that earlier. So, and they actually, and if And if we think about birds and crocodiles, as independent species, they actually share many similar features. For example, they both have four-chamber hearts. They sing to defend territories and attract mates. And we're not talking about crocodiles going A B C D. That's not like that. The way crocodile sings is very different, but they do make some vocal means to defend their territories or attract mates. And then birds and crocodiles both build nests and care for eggs by warming them. Okay. And of course, we know birds warm their eggs by sitting on them, hovering over them. Crocodiles won't hover their whole body over the eggs because they're huge. So usually they do it with their neck, or they actually put some kind of covering over their eggs and warm their eggs that way, which is pretty interesting, I thought. Hence, if we use the approach of phylogenetic bracketing, biologists predicted that dinosaurs should also share these same features because dinosaurs are the closest sister taxa, specifically theropods, are sister taxa to birds. Unfortunately, for internal um, for internal organs such as the hearts, they rarely fossilize. So there is no way to compare the hearts of dinosaurs and birds. And of course, it is impossible to test whether dinosaurs sang to defend their territories or attract mates because they no longer are alive for us to observe these behaviors. So we can only look at the other two features that birds and crocodiles share, which is building nests and caring for their eggs. We can actually see that in fossils, at least indirectly see that. Here, there are two fossil discoveries that have supported these two particular traits. So first, a fossil embryo of a oviraptor, okay, dinosaur, was found inside its egg, which means whatever egg fossil we see should have embryos inside of them. And second, they found fossil of an oviraptor crouching over a group of eggs, similar to how birds kind of sit on their nest today. So here is a picture of that particular fossil where it shows that the orientation of their bones of this dinosaur surrounded and covered the eggs. Here are the eggs, here's the front limb, here's that hind limb, and it's kind of like covering and hovering over the eggs. And they say that it probably, kind of this fossil suggests that the dinosaur died while incubating or protecting their eggs. For these fossil records, top artists have actually reconstructed the dinosaurs and drew this particular picture with the dinosaur's posture sitting on his nest, as shown in this particular picture here. Which leads to all these pictures we see of dinosaurs. We cannot visually see this is how dinosaur look like. Whatever models or pictures, movies we see of dinosaurs are what we think they possibly can look like based on the fossils we see. Okay, that's how we go, have these huge skeleton bone structure of T-Rex in a museum. Because it is due to the fossil records that we're able to deduce this is how big they are. This should be how they look like. Okay. Again, I must emphasize that this is what science can infer based on fossil findings. Remember the word infer. We can never actually see anything like this happening because dinosaurs no longer exist. Unless somehow in the future, who knows, centuries later, time machine was invented. I don't know, okay? But you see these fantasy movies all the time. So it is not a direct observation of dinosaurs sitting on their eggs that we're seeing in this picture right here, but an inference based on the best data available that we have, which are fossil records, and we deduce that, or scientists deduce that they kind of look like this, sitting on top of their eggs. So thus, they say that fossil records discoveries of nests and parental care in dinosaurs provide data that suggests the hypothesis is correct, and the hypothesis being the phylogenetic tree. And they have found multiple other types of dinosaur fossils that have this similar pattern where they're hovering over their eggs to show that it is very likely that they used to sit on their eggs, and sort of hover over their eggs this way to warm them up, just like birds and crocodiles do. And thus, fos oh, excuse me, as a way to set the stage and talking about plants and animals, we're finally done talking about phylogenies and phylogenetic trees. So, but we'll keep coming back to this overall evolution, natural selection throughout the whole semester, because that's the whole theme of Bio 1407, second part of biology. But, this is, this last part of the chapter kind of talks about kingdoms and domains, and we will be talking about plants and animal species in the next chapters for the rest of the semester. So the last section of this chapter talks about how biologists have changed classifications over time. So this is the best way we set the stage going into the organisms that we'll be talking about. So they once classified all known species into just two kingdoms, which are just plants and animals, until this later transition into classification of five kingdoms: Monera, which are the kingdom of prokaryotes, including bacteria, and then we have Protista, which are unicellular organism, and then plantae, fungi, however you say it. I've heard it both ways. There's fungi. There's people that say it's fungi. I always call it fungi, so I'm going to call it fungi. And then you have Animalia as the fifth kingdom. This division into five kingdoms actually have set prokaryotes apart from all other eukaryotes by placing them into their own little kingdom called the Monera. So they also have discovered that prokaryotes actually differ from each other as much as they do from eukaryotes, because they used to say, oh, prokaryotes are so different from eukaryotes that we're going to set prokaryotes apart in their own little kingdom. But they're not little. There's a vast, diverse species of bacteria that we know and prokaryotes that we know. So that's why they found that, hey, that's not the only thing that's there. Prokaryotes are actually also very different, vastly different from each other, just as they are different from eukaryotes. So biologists then had to adopt a three-domain system. This is why domain was added later on into the Linnean classification into bacteria, archaea, and eukarya. Okay, these are the higher, the domain system that is higher than the kingdom level. Okay, which is why there's a D before the K now. Let's see. Okay, so domain bacteria usually are mostly made up of currently known prokaryotes, as I've talked about. The domain archaea is actually a diverse group of prokaryotes that inhabit wide variety of environments. Okay, why they came up with this domain archaea is because they used to think that bacteria cannot live in very extreme environments, like in hot springs that are really, really hot, or in really, really extremely acidic environments. But they have found out that they're incorrect. There are prokaryotes that can survive these extreme environments, but there are specific prokaryotes. This is why they came up with the domain archaea, because they're so different from the usual bacteria prokaryotes that we now know nowadays. Okay, so that's why they came up with this whole domain of very interesting specialized prokaryotes that can inhabit these extreme environments. Okay, and the last domain, eukarya, then is all organisms that have cells containing a true nuclei. That's what eukaryotes translates to or define as anyway. So if we look at the three domain right here, domain system, phylogenetic tree here, we can see that the history of life mainly is made up of single-celled organisms, because the only ones that are multicellular on this list of species right here are actually highlighted in red, which are plants, fungi, okay, or animals. Only three of them. Whereas all the black ones that you see on the list here are actually all single-celled organisms. So the kingdom Monera is not really, appropriate, because it would have members in two different domains, some in the bacteria domain and some in the archaea domain, because they're both prokaryotes. This is why eventually that, biologists think, okay, maybe the kingdom Monera is not really appropriate. They call it obsolete. I don't know, but it is still till this day considered as one of the kingdoms. I don't know, maybe in the distant future they would completely remove it. Like I said, science may keep changing, just like how we used to consider Pluto as a planet, and now we say, oh no, it's not considered a planet anymore. When I was in elementary school, I learned Pluto was a planet, but now it's not. So that's how science keeps changing. But they're arguing that Monera is not a good enough kingdom because of the fact that they have prokaryotes residing in two different domains. And in addition, the kingdom Protista is also something biologists have argued about because it includes members that are closely related to plants, fungi, or animals than other protists. Like if you're in the same kingdom protists, you're supposed to resemble each other as protists. But they say that most of them are actually more resembling plants, some are resembling fungi, and some are resembling animal cells. So they're not resembling each other. So that's why the kingdom Protista is also one of contradiction, because we don't know if we should keep it or not. For now, it is still a kingdom anyway. But who knows 10 years later what this has become. So as you can see, classification is not a straightforward type of deal. It continues to become more complex as we make more discoveries. Hence, new research continues to change our understanding of the tree of life. I mean, who knows that most of our history on this particular phylogenetic tree is mainly made of a single-celled organism, okay? As that's what it says here by this phylogenetic tree. Only 3 of these are multicellular, so maybe our history has a lot to do with the unknowns of these prokaryotes doing something and eventually evolve to become eukaryotes. Don't know. That is something still in the process of research. So according to the phylogenetic tree of 3 domains, eukaryotes and archaea are the more closely related than either is to bacteria. Because you can see here's a common ancestry for this split here, and then the common branch point between eukarya and also archaea. And this common ancestor goes with bacteria. So that's why these 2 domains are sister taxa of each other, and they are both more distantly related to bacteria. That is what the phylogenetic tree says, okay? Well, turns out that some genetic studies have found that there are genes related to metabolism in yeast cells, and yeast cells is considered a unicellular eukaryote, and they say that these particular genes are actually more similar to those in the domain bacteria than archaea, which kind of contradicts with the fact that we just said eukaryotes and archaea are more closely related than to bacteria. So this suggests that, that eukaryotes may share a more recent common ancestor with bacteria versus archaea. This is contrary to the original hypothesis of the phylogenetic tree here, okay? But we have found otherwise. So why is there this inconsistency in our phylogenetic tree? Well, the comparison of complete genomes, because that's what biologists or systematists have to come down to, is sequence the complete genomes of the 3 domains, and they show that there were actually substantial movements of genes between the 3 domains. These movements actually took place through what biologists or systematists are calling it horizontal gene transfer, which is actually a process where genes are transferred from one genome to another through mechanisms such as exchange of plasmids. And there's also transposons. Okay, we're not going to talk about that. That's a genetics thing. So I kind of omitted that out of this in case you're like, what are those transposons? We'll talk about that if you click genetics. Okay. And then, so just in plain terms, DNA, exchange of DNA sequences, viral infections, and fusions of organisms through endosymbiosis. You may have heard this theory from Bio 1406 that 2 cells somehow end up living with each other, eventually fusing to become one cell, okay? And that's a whole theory about how prokaryotes become a eukaryotic cell, because a mitochondria used to be a bacteria and now it lives in this endosymbiosis with the actual cell itself and became one eukaryotic cell. That's a whole theory, again, of endosymbiosis. Again, it's a theory. We don't know if it's true or not, but it's the best explanation we've got. So, um, other than passing genes vertically from one generation to the next in phylogenetic trees, we've been showing you all along throughout this chapter, they say that horizontal gene transfer must have also occurred, where prokaryotes were able to pass genes to eukaryotes. And that's why these yeast cells right here have metabolism genes that are similar to prokaryotes in the bacteria domain, compared, more than so in the archaea domain. And then another example in this horizontal gene transfer that show us this is true is that there is this algae called Goldiaria sulfuraria. Okay, this particular species can amazingly survive at very highly, or you can say extreme conditions, such as highly acidic environments or extremely hot environments, or in environments where there's high concentration of heavy metals that most algae can't live in. So these are environments that, like I said, most things, not only algae, come to think of it, most things cannot live in. So researchers have found that these specific genes that these algae have are transferred from prokaryotes that have enabled these specific gene species of algae to thrive in such extreme habitats. Hence, overall, biologists says that horizontal gene transfer played a very key role throughout the evolutionary history of life, that they continue to do so today. This is why this chapter wraps up with this horizontal gene transfer concept. Okay? So other than those vertical changes, we're saying that here are these two descendants sharing common ancestor. They're saying, oh, there's gene transfers horizontally as well, not just vertically. And that's all I have for you for chapter 26 regarding phylogeny and the tree of life. Again, note that I did skip over a few sections on cladistics. The topic of cladistics, shared ancestral and shared derived characters, coming up with your own phylogenetic tree with derived characters, maximum parsimony and maximum likelihood, genome evolution, molecular clocks. All these topics are actually overly technical that we don't need to be covering them in detail. As you can see, we are at 1 hour, 6 minutes, so that our lecture is pretty long as it is. So if we go into those detailed topics too, it will take probably another hour to talk about them. So that's why I'm going to skip over those sections. So make sure you're aware of that while you're doing your homework. So if you want to read about them, though, you surely can. They are from your textbook, okay, from chapter 26. And I've already listed those particular topics earlier, just now, by talking through them. So let's go over these practice questions and we will be done. All right, which of the following taxonomic categories includes the fewest number of species? It is Panthera because it is a genus compared to the other that are order, class, etc. Genus is second most inclusive compared to species. Okay, animals that possess homologous structures probably blank. A, evolved from a common ancestor. That's the whole point of our definition of homology or homologous structure. They have to have been from a common ancestor. It can't be because they were in similar environments. Which is the correct way to write the scientific name for humans? C, Homo sapiens, being italicized for the whole word and also capitalized for the genus portion of the scientific name. Which broad taxonomic group is not a domain? C, Plantae, because there are only 3 domains, which are bacteria, archaea, and eukarya. Now we're looking at phylogenetic trees. According to this particular phylogenetic tree, which is the closest relatives of the group composed of cetaceans and hippopotamuses? D, other even-toed ungulates, because here's hippopotamuses, here's cetaceans, so this is a group right here, and they share a common ancestor with other even-toed ungulates. Identify the sister taxon to seals. So don't be afraid to draw things on these phylogenetic genetic trees in order to read them correctly, just like I did here, okay? All right, so here are seals. So the sister taxon will be the one that's connected directly with it, which are bears. According to this tree, which is the most distantly related to the other even-toed ungulates? Here is other even-toed ungulates. So here's cetaceans, here are bears, and here are hippopotamuses. Which one is the farthest in terms of common ancestry terms? Okay, well, it would be bears, because this is the common ancestor between ungulates and the lineage of bears, whereas this is a common ancestor between ungulates and hippopotamus, and etc. So that's why this would be the most distant one if we're only comparing between these three species. Okay, you discover two trees that each have thorns to protect them, are from herbivores, but they are in different classes of plants. So, but these two trees both have thorns as a trait that they share. And many non-thorny plants are actually between them on the tree of life, when we're talking about phylogeny of this particular two trees. So what has happened? Why are they on completely different lineages, but yet they share the same trait as having thorns? Well, most likely they have what's called convergent evolution, where they both were put under similar environments. So there's these herbivores that are trying to eat them. So they have evolved by natural selection in both cases to have thorns, not because of a common ancestry, closest ancestry case, but because of environmental driven things. So we can say that these two trees have an analogous trait such as thorns. Okay, they're not homologies or homologous structures. Now you are studying a strange new organism. Examination of its structure show it to be truly multicellular. Where would you begin to classify it? The fact that it says multicellular tells you that it would be in the domain eukarya, because archaea and bacteria are both single cell organism. We can't go to the plant kingdom yet because we just know that this is a multicellular organism. We have no other information. So we start with eukarya. Which of the following could be homologous between two taxa? All of the above. Because when we're talking about homologous structures, it can be bones, it can be limbs, it can be leaves, it can even be genes, molecular homology. So that's why all of the above can be homologous between two taxa as long as they are sharing the same common ancestor. And that's all I have for you.